Hello and welcome to Nature Live Online, the show that takes you behind the scenes at the Natural History Museum, London. From dinosaurs to DNA, from volcanoes to vampire squid, we bring our museum science direct to you at home. Now, today we're talking DNA because the history of every organism on our planet is recorded in its DNA. So, what might we learn if we could read the genes of all those plants and animals? Well, the Natural History Museum is currently working on an incredible project that aims to sequence the genomes of all UK species. So, to find out why and how we're achieving this feat, I'm going to be chatting with two museum scientists involved in the project, Inez and Laura. Now, I've got loads of questions for them, but as always, this is also your chance to put your questions to our scientists. So don't be shy. If you've got any comments, any questions, post those in the chat. We'll do our best to answer those for you. Say hello, tell us where you're watching from. We'd love for you to join in the conversation. But let's now welcome to the screen our two guests for today, Inez and Laura. Are you there?
Yes, I am.
Hi, Alison. Brilliant. Hello, both. It's brilliant. It's fantastic to have you both on the show today. I can't wait to get into this incredible project. So let's dive straight in. It's called the Darwin Tree of Life. Inez, what is this project all about? Who's involved?
Well, it's an incredibly exciting project based, a broader project at Sanger, which is the Tree of Life project, which is basically aiming to sequence the whole genomes of every eukaryotic species in the UK, which amounts to some 70,000 species. So it's a huge, huge project, really ambitious. And of course, we're not doing it alone. NHM is mainly focusing on collecting and sampling marine and terrestrial invertebrates. So we are branching out a little bit at the moment. As you can see on the screen, there are numerous large biological organizations, everyone involved in the collection to get all the species for the project. But ultimately, all the magic happens at the Wellcome Sanger Institute, which is where the whole genomes will be generated.
It's an incredible set of collaborators there. What's the museum contributing to the project?
That's a great question. Oh, sorry. That's a great question. So the museum is contributing many different elements to the project. For one, we have expertise in identification. The museum employs over 300 scientists, many of which are taxonomic experts specializing in the identification of specific groups. And a key part of this project is assigning the correct taxonomic names to each specimen. We also go out and collect the specimen. That's the fieldwork, targeting our high biodiversity hotspots to maximize the diversity of the specimens collected. And then finally, at the museum, we have our specialist labs, the molecular collections labs. These have our freezers in our liquid nitrogen tanks, which are all really important for DNA preservation.
That's absolutely brilliant. And what are your specific roles in the project? Inez, if we start with you.
So I'm the sampling coordinator for the Natural History Museum, which means that I basically coordinate all the samples that are collected either through fieldwork or through external submissions for the project. So it means that I make sure they come to the museum in the right condition, they're processed accordingly, and then, of course, shipped off to Sanger in the right way. A part of my role also involves making sure that all the specimens that come through are curated appropriately. Since we are a museum, we have a responsibility to keep a record of all these specimens. So making sure there's a good physical record and a good digital record as well.
And Laura, what do you get involved in?
Yeah, so I'm a research assistant on the project. So my work primarily involves sampling and preparing the specimens for DNA barcoding and whole genome sequencing. This involves fieldwork, taking pictures of the specimens and then working closely with museum curators and biological recording schemes and partners throughout the UK.
Very cool, very cool. Now to say that this is an ambitious project is a little bit of an understatement, isn't it? 70,000 species that we know of in the UK. Why attempt it at all? What can having a whole genome tell us?
That's a really good question, Alison. Well, mainly part of it is just obviously interest. We do not have the whole genomes of a large majority of these species at all. And of course, the more you find out about the species, this also leads to how better we can preserve it. And it's also incredibly important that this project is contributing to how we measure biodiversity. As we know, biodiversity is a sign of ecosystem health. The biodiversity in the UK has been decreasing steadily in the last 50 years or so. So we see this project as acting as a snapshot of what we have in the UK at the moment. And by archiving and storing these specimens appropriately, we will also be able to preserve them for future research. So I think it's really, really important we have a record of what we have. And this also allows us to push technology further. Like mentioned, if a lot of these species haven't been sequenced, what more can we find out about them, and also help us in how we can preserve them for future generations?
Absolutely. And at what stage are we in the project? What's been done so far from the museum side, Laura?
Yeah, the project began in 2019 with a large collecting trip to Ainsdale. I began working on the project in 2020, and to date, we've collected over 1,900 species, and that's across 480 families. So it's going well.
That's pretty impressive. How do you decide where, but more particularly, how do you decide what you're going to collect? Inez, is that your job as a sampling coordinator?
Well, it's an incredibly kind of ambitious task in itself, because there wasn't actually a record of every single species in the UK, kind of a definitive list. So a large majority of the initial stages of the project was trying to compile a list using kind of recording schemes or kind of various hobbyist groups which keep a record of the species they come across in various areas of the UK. In terms of what to collect, we are still in the pilot stage of the project. We are aiming to have a family representative from every single species, but this is still 2,000 species in itself. However, in our collecting, we do just generally aim to collect as much as possible, but it is very targeted. So we are definitely trialing the technology that we have on these samples first, and then hopefully in the next stage of the project, we can move on to expanding our search for the other species on our lists.
So it's there's flexibility built in, is there?
Well, there has to be, because as you mentioned, there never was a definitive list. This is the first time that's been trying to be compiled. Include things like invasive species. Do we include those? How established are they in our UK population? So this project is very much evolving, and even in its initiation kind of in early 2020, I think so much has changed the more we learn about the species we actually have in the UK in itself.
Absolutely. And Laura, tell us a bit more about the collecting that you've been doing so far, because I imagine the pandemic had put a bit of a spanner in the works. How have you been collecting?
Yeah, absolutely. So once we have the species list, as mentioned by Inez, we narrow down the locations, and then we choose a site in which we're likely to encounter our target species. And then we have to pack our kit. So it sounds like an easy process, but it actually can take quite a long time. So to ensure that the DNA of specimens is well preserved, we maintain the cold chain throughout the process. That's from collection to whole genome sequencing. And specimens are either stored on dry ice, which is about -80, or liquid nitrogen, which you can see here. That's -200 degrees. And a dry shipper is a crucial piece of kit. So I fill that with liquid nitrogen, and that can take between 5 and 10 minutes, depending on the temperature outside.
Wow.
Yeah, and then also, so that's kind of the setting up side of things. Then when we move on to the actual collecting in the pandemic, so like many areas of life, we did have to make adjustments. So we did change our sampling technique. For example, we work outdoors in socially distanced pop-up labs where specimens are collected and fully processed on site, ready for DNA barcoding and whole genome sequencing. Additionally, we process samples at home, setting up mini labs in our houses and processing these samples to contribute to the DNA reference library in the UK. Finally, we have small group collecting. This was surprisingly and really important during the pandemic, which of course we're still in. We took part in small group collecting. So these are bubbles, and we have a wonderful network of collecting partners and taxonomy experts throughout the UK. And they've been critical to the success of this project. And then this year, we've been able to go on several larger field trips, which we're really pleased to do. So one such field trip was Millport. This is an island located off the west of Scotland, and we were there for 6 days, sampling the rocky shores, the seaweed, and the ocean floor. A total of 192 specimens were collected for DNA barcoding and whole genome sequencing. And this includes species that were collected by museum scientists in 1860, and they sit next to them on the shelves now at the museum.
That's absolutely brilliant. And it's fantastic how much you've been able to do, even in spite of the pandemic. You set up those labs and Brilliant. I'm so impressed. So you collect your specimens. What happens next? Do they come back to the museum and then what happens?
Yeah, so in order to preserve the DNA, we're working on the cold chain in this project. So identification is occurring mainly in the field upon collection where possible. And then we come back to the museum where Inez will take over.
Yes. So, well, as Laura mentioned, maintaining the cold chain is actually a really crucial part of the project because we have to maintain the DNA. It's worth noting at this point, we've been talking about whole genome a lot. So the aim of the project, of course, is to generate the whole genome, which means all of the DNA that you have in your cells. But there is also technology that allows us to only sequence a fragment of that DNA, which is called DNA barcoding. All of the specimens we collect do go through a DNA barcoding process as well. And this is mainly just to act as like an identification confirmation, because there's lots of DNA barcoding archives around the world where we can kind of compare our results. However, it's worth noting that, like a lot of things in this project, it hasn't been done before. So we are coming across lots of species where there is no DNA barcoding record. However, once they pass the DNA barcoding stage, so as you can see in these plates, they're processed in ethanol. So we take a chunk of tissue from each specimen. So you can see these are clearly some arthropod legs, and those are processed in the lab here. And then we kind of generate a large list of identifications based on their short DNA. However, for Sanger, they want the whole genome, which means that the specimen has to be kept in -80 throughout to preserve the DNA. So when they get to Sanger, the whole genome can be generated there. So here's some liquid nitrogen tanks, which are actually around minus, I think, 160 degrees. However, it's been found that they don't need to be maintained that cold. But if we do want to keep them for future generations, we're talking 200, 300 years into the future, this would be the way we should be storing them and that we're aiming to store them.
Fantastic.
So, yeah.
We've actually had a question that come in from Mariel on YouTube asking, how are you managing to find available sequencing machines when they're all busy sequencing SARS COVID?
Well,
another pandemic related question there.
Yeah, well, that's a very, very good question. Luckily, I believe our samples aren't as high as perhaps the COVID submissions across the UK. It's worth noting this project was set up before the pandemic happened, as Laura mentioned. We've had to adapt in huge ways to do any kind of work on this project. So there is funding generated and set aside specifically to sequence our samples. However, we're not obviously taking away from the research that is done around COVID currently.
No, and we have in-house sampling and sequencing machines. So the sequencing machines are found at the NHM and also at Sanger. So they're separate from the COVID ones.
That's
very separate.
Yes, absolutely.
That's good to know. And a great question there from YouTube. Keep those questions coming. We are here to answer your questions too. So pop any questions you have in the chat and we'll answer those as well. So we're doing the barcoding. Sanger is sequencing the full genomes. Do we have any full genomes successfully sequenced so far?
Yeah, we do. So there've been several, but one of them is this charismatic example. So it's a fly called Bibio marti. It was collected by a museum colleague in May 2020, and the genome has been published in October 2021. It's a common and widely distributed species in the UK. And so you've probably seen it yourself. It's typically found in grasslands and at woodland edges. And the adults have a really short life cycle, but the larvae the juvenile stage is much longer, and they'll feed on rotting vegetation and subterranean parts of plants.
That's brilliant. Great that it's a museum specimen as well. So what might we be able to find out from having this fly's whole genome?
Well, firstly, it's worth noting this is in terms of an NHM collection. This is NHM's first whole genome. Like Laura mentioned, various other partners have produced whole genomes in the past, but it is quite special that someone who worked on the project submitted the fly, and this has gone through the entire process from collection to the whole genome sequencing. As we mentioned earlier, in the end, having the whole genome, which is publicly accessible, by the way, so anybody can view this across the world, it's an incredible resource. And whilst this fly in particular was found in the UK, who knows what kind of knowledge you could find out about it if whole genomes from the same family are also generated, not just in the UK, but across the world.
Absolutely. It's very, very exciting. But we've had another question from one of our viewers, Noah. Hello, Noah. He's a regular viewer, age 7. He asks, what's the most common species that you found that you've been finding? Laura, this might be a question for you.
Oh yeah, that's a really difficult one. So lots of species are really common. One of my favorite ones that we get in the summer months is the southern hawker. So it's maybe not the most common, but it is really charismatic. So I recommend looking out for that. But in terms of most common groups, we get lots of beetles, lots of flies, and lots of wasps as well. They tend to be the most common. Yeah, thank you for your question, Noah. That's really good.
Yeah, it's a great question from Noah. Are you finding anything surprising when you've been out collecting? Anything you didn't expect?
Yeah. Well, yeah, go on, Inez.
Well, I was just going to say from personal experience, there is a lot of rhetoric that summer or spring is the busy season where all the insects are out. But of course, during the winter, that's where the kind of more cryptic species appear. So we're talking about the woodlice, the worms, the millipedes, the centipedes, sort of kind of the less obvious or maybe the less flashy inverts. So for me, it's been really interesting to see the kind of seasonal differences in what we're finding. And of course, during the winter, they're a lot harder to find. So that's been quite an interesting challenge for me.
And Laura, anything to add?
Yeah, well, personally, one of the most strange things that I found was when we were collecting in Millport, and I didn't realize how big worms are under the sand because I'm not a marine specialist. So yeah, that was fantastic. I was really tiny little hole on the sand, and then you dig down a couple of meters and a worm about this big. So yeah, that was really, really cool.
And we've had another great question from this time from Facebook. Pauline is asking, is this project being done simultaneously internationally as well? And is there a central database?
So this is the first project of its kind specifically based in collecting all the species in one country and sequencing them. However, there are lots of other projects that are looking primarily at barcoding, so the shorter section of DNA. And we hope that this will be a pilot project and provide some guidelines for other countries that may wish to endeavour on similar projects.
Yeah, it's worth noting the DNA barcoding aspect isn't new technology at all. So there is kind of international barcoding schemes out there. I do believe that they are planning to do more of this in various different countries because, like I mentioned, measuring biodiversity is becoming a really, really important kind of data set to have, especially in the current climate. So it is really, really exciting to see how we can kind of contribute the knowledge that other countries might be able to utilize as well.
Yeah, we know DEFRA are doing a lot of work on DNA barcoding in the UK, and then there's a new project coming up called UK Barcode of Life, which is in collaboration with Europe and other countries as well.
Yeah,
brilliant.
And if Pauline's interested in looking up DNA barcodes, there is an international database called BOLD, which means the
Barcode of Life database.
Barcode of Life database. Barcode of Life database. But that's an international one. So if she wants to look up any of the DNA barcodes that have been submitted worldwide, that would be a very good place to start as well.
Yeah, and lots of sequences are freely available in there too. So it's a great place.
Yeah, I mean, I guess once we have the barcode, the reference, the DNA references, and the full genomes, potentially anyone can use them. So yeah. Very exciting. Lots of potential collaborations. But with all of these new cutting edge techniques, these DNA techniques, I'm also thinking things like environmental DNA. Do we still need traditional taxonomy?
Yep, well, that's a really interesting question, Alison. And the answer, I think, is definitively yes, we do. Although kind of the origins of taxonomy were very much based on morphology. So as we know, as it was kind of set up in the 1700s by Carl Linnaeus, they didn't have the technology we have now. So taxonomy in itself has changed a lot in the last few hundred years. But I think it'll be unfair to completely dismiss all the work that has gone in from categorizing these specimens from kind of a morphological viewpoint. If anything, our project is adding to the knowledge and is requiring even more taxonomical input to be able to increase the amount of knowledge we have in kind of, all the species we have around us. I also think it's important to mention that science is something that constantly changes as the technology changes. And I think it's really important to be adaptable. And as a museum, which was based on collection and was based on kind of taking snapshots of the species we have around the world, it's even more crucial that all those skills are kind of aligned as we kind of head to, learning more about how we categorize specimens based on not just morphological, but evolutionary data as well.
And they're absolutely complementary. I think that we need to stress that as well. So you can get species that are actually genetically very similar, but they are separate species. The behavior is very different, and you can use morphological differences in the specimens to look at this and assess this. So they have a nice interplay with one another.
Absolutely. Our collections are still definitely relevant. That taxonomy is still relevant, but we're building on it, aren't we? So some of the things that you collect come into our collection, do they? They come into the molecular labs, is that right? The freezers?
Yeah, they do. So our collections, as we were saying, they're hugely important, and the collections are used in several different ways. So our existing collections can be used to identify species which are coming in, specimens that are maybe more difficult to identify. And these are matched to the identified specimens in the collections, the reference collection. And we're continuing to build the collections. So specimens in this project go into the main collections, vouchers are incorporated, and in that way, we enhance the collections in terms of the voucher specimens, but also in terms of data. We have the DNA barcodes and the whole genomes. Then in addition to this, we have a new type of collection, and this is where we have the frozen archive of material, in which we're bringing together two aspects of taxonomy, the morphological side of things and also that genetic data, as we were talking about before.
It's such a rich source of information, both of those together. It's absolutely brilliant. We've had a great question from Timothy come in, aged 9, on YouTube, asking how much DNA barcoding have you done so far?
Yeah, so that's a fantastic question. Thank you, Timothy. So far, in terms of species, we've covered it's 1,945 species, specifically collected by the NHM. The other partners have collected different and more samples than that. And that's across 480 families. But in terms of the actual species count, so the number of specimens that can be duplicates of the same species, that's closer to 6,500 specimens. So it's been a lot of work, but it's going really well. Thank you.
Yeah, pretty impressive. Lots more to do.
Oh yeah,
I haven't quite got to the 70,000 yet, but we're getting there.
Yes. And about 24,000 of those are insects as well. So slowly, but
we're crawling there.
Yes.
Yeah. It's absolutely brilliant. So I was actually going to ask what's next for you in the project, but actually Artie on YouTube has preempted that question, asking what field trips have you got coming up, and which ones are you looking forward to? Have you got any plan for the future?
Yes. So, we sampled in Scotland this year, as Laura mentioned. So we did a marine trip, but we've also done terrestrial sampling as well. For next year, we're definitely planning to go back to Scotland. It's an incredible, obviously not just an incredible place to visit, but there's so many really interesting highland species there as well. And since now the pandemic, although it's not over, is sort of loosening up slightly, it means we have access to all these areas that we didn't before, especially things like joining bioblitzes, which tend to be kind of more locally run, where, we have lots of people involved in the sampling kind of cause. So we are planning to join some of those in the Broads and more kind of East England, and potentially some in Wales as well. So fingers crossed we get to do lots more sampling next year.
Yeah, and that should ramp up in springtime and then continue kind of throughout until the autumn, when we'll tail off again.
That's brilliant. Absolutely brilliant. And so do we think that this is what collecting looks like for the future, would you say? Traditional taxonomy plus DNA?
Well, it's interesting you mentioned eDNA, because part of the reason why we're doing this is by having a much larger DNA barcode reference library, as Laura mentioned, it really kind of increases the potential we have for doing environmental DNA sampling, which if the viewers aren't aware, is when you are able to kind of measure the presence of a particular species just by using kind of very small samples, such as kind of like a scoop of water or a leaf, and a bit like kind of adding in a forensic element to it without having to actually see the animal. You can detect its presence there. And this is actually obviously, hugely encouraging when it comes to any kind of biological sampling. If we don't actually have to see the animal and we know that its presence or absence, who knows where this will take us, really.
Yeah, absolutely. It'll continue to be that complementary blend between the two going forward. I would definitely expect to see that. And in terms of the cold chain and preserving DNA, there's so much work being done behind the scenes to make that easier. So you can have different preservation methods, but still get high quality DNA. So I'm personally really excited to see where that leads us in the future.
Fantastic. So what are your hopes for the next 12 months, Laura? Anywhere that you want to go?
Yeah, that's a big question. No, I like that. Yeah, I'm really excited about the Welsh field trip that we have coming up. There's several different places we want to go to and different habitat types in Wales. And we haven't had a chance to get there yet due to various reasons, including, travel restrictions. So definitely looking forward to that. And looking forward to seeing what comes out of the data that we're producing as well. The whole genomes, they're being published at a faster rate now, and it was really exciting to see what's going to happen in the next 12 months, but also beyond that into the future.
Yeah, Laura mentioned, could we include the Bahamas in this? And I said, I don't think that is included in the UK. But
I think it's important that we compare the differences in genomes between any common species that we might have. Maybe.
I like your thinking, Laura. I do. Nice try. And if people are interested in finding out more about the project and finding out what's been discovered, what genomes are being sequenced, where can they go? Can they find out online?
Yep, so there is actually a Darwin Tree of Life data portal that is run by the Wellcome Sanger Institute. So you've just typed that into Google, and that has a complete list of everything that has got to Sanger at this stage, as well as there's tabs that show you kind of its process in the from the actual getting to Sanger to having its whole genome produced. It is actually worth mentioning as well that not everything we collect does generate a whole genome for various reasons. So therefore, this is why we have to often keep really accurate records of what we have. So if anything does need to be sampled again, or we need a better specimen, we're able to do this because we're still very much in the trial phase. But it's been really exciting to see how just the technology has improved in the last 2 years in the amount of DNA we can get out of a particular specimen as well.
Absolutely. And we also have a blog post as well up that gives you a bit more detail about our collecting trip in Millport. So if you head over to NHM blogs, you can have a look at that too.
Brilliant. I would highly recommend that. It's a fantastic project, very exciting, and I can't wait to see what happens next. But sadly, we are out of time. Thank you, Inez and Laura, both of you, for joining us today. It's been brilliant chatting to you, finding out all about this fantastic project. You'll have to come back and update us as it goes along, because I'm sure I want to hear about all this exciting field work coming up in the next year. So we'll look forward to hearing from you again, but we will say goodbye to you for now. Thank you again. Thank you so much.
Thank you so much.
Thanks, Alison. See ya.
Take care. Bye.
And thank you to you, our viewers, for tuning in today, for all of your fantastic questions. We always love to hear them. Check out the museum website. Check out the Darwin Tree of Life website as well for updates on the project. It's well worth doing. And do remember to join us again for Nature Live Online next week. We're going to be finding out why we should all love lichen. Until then, take care. We'll see you next time. Thank you.